So good day. Today's lecture video is all about flora and fauna in freshwater ecosystem. So this is part 3 of our lecture for week 7 to 9. It's all about biological sampling of the environment and its sampling method. So now let me show you the flow of presentation for this video lecture. So we will be tackling about planning of research, identifying sampling sites, collecting samples and pre-treating them. So we'll be talking about collecting samples in the plankton community, the benthic community, aquatic macrophytes, periphytic algae, and fish community. So let's now know how to plan the research. First is when you deal with freshwater and coastal researches, no, you need to state clearly the objective for doing the assessment. So, no, nauni mo kung ano mag-study ka ani, what for? You don't only study aquatic or freshwater and marine ecosystem just for the purpose of doing something kanang overstudied na kayo, but maybe you want to relate it as bioindicator if this species are bioindicator of the environment. So when we say bioindicator, so if for example they know if for example certain what they call this changes in the environment now occurs, so this species may dominate in the area or may decrease their population in the area. So that's what we call bioindicator species. So kung for example kung ugaw kaayo ang environment, for example, and these species are known to be bioindicators. So let's say the domination of their population may suggest that the quality of the water is kanang hugaw, for example. So definitely these bioindicator species are what we call tolerant species, no, that would tolerate to the changes in the environment. So of course you need to state clearly what your objectives are. So second, get a topographic map of your study. So definitely when you deal with rivers, so for example rivers now, river has a one pathway, so which means you'll be dealing with upstream, midstream, downstream to know the differences. So with that, no, you need to get the topographic map. So definitely you'll have to you'll be given an idea, no, to what extent are you going to, or what scope your study is all about. So third is designing of the sampling scheme. So first things first is when you go to your sampling site and when you deal with coastal or even freshwater, or marine ecosystem studies, then definitely you have to do a sampling before that, your pre-entry. So with pre-entry, you need to do, you need to give letter to the local government or barangay captain to do a research study. Then then after that, you'll have the scoping, no? So you need to do site visit, ocular inspection. So definitely kung asa ka mag-start sa imong sampling, asa ka mag-end, unsay scope lang sa mong sampling, no? So definitely that's how you plan your sampling scheme or how you plan your sampling period. So if your study will only focus on that particular barangay, then you stay on that barangay. You create sampling plots or sampling points within the barangay, or maybe if the whole area uh, scopes 3 barangays, then definitely you'll have to do ocular visit within the 3 barangays for you to know whether, well, when you will, I mean, what barangay you will start and you will end. And kana siya no kung unsay relationship between the 3 barangays. So you don't only pick those barangays because it's the scope of your study, but pick those barangays because they have different definition of sampling sites. Like for example, if barangay, uh, let's say Poblacion, is known to be a recreational area because it has different beaches, no, and resorts, uh, which can be seen, no, evidently seen in the area. So other Loyong Bunbun that would be considered as your residential area, for example, because nearby, no, there are people living nearby the coastal areas. So nana no, dapat ang definition ninyo sa kada site ninyo should be defined clearly in your sampling scheme. So another part of planning the research is the reconnaissance survey. So it's still the same with ocular visit, reconnaissance survey or ocular visit, site survey. Those are examples of uh, how you've done your sampling scheme. So after that, you need to know whether this part here should be included in your study, or maybe uh, that will be your limitation. That will be part of your limitation. So depends on how you survey your um, capacity in terms of uh, the limit, the time limit that is given to you in your study, time frame, I mean. So also uh, the finances, no, that will be available to your study. So if your study would be that costly, then definitely you have to limit, or yes, you have to limit your study, no, based on your capacity. So fifth is the collection of biological samples. So when you deal with coastal and marine ecosystem studies, then definitely you have to collect samples, no? Maybe in terms of species or in terms of, let's say, um, bacteria, maybe in the water. So that's it. Another is second, after planning the research, you have to identify sampling sites. So, of course, like I mentioned a while ago about site identification, you have to determine whether this particular area, you know, how you define that. So maybe, for example, you define that as recreational area, industrial area, maybe residential area. So you have to identify, you have to operationally define your site, not when you identify your site. So another is observe existing microhabitats. So this one you need to consider in your sampling sites katong mga minute information. So examples are, let's say this sampling site is, what you call this, is part of this kanibitang, for example, in Luyong Bunbun, no, kana nabantayan ninyo ng dapit na San Miguel nga distributor. They are not in an industry, but they are just a distributor here in, I think, Cagayan de Oro, Misamis Oriental. But as you can see, if you try to notice the area, no, kanang where siya ginadak siya o kanang mga private bitaw nga yacht. So then na gina-park. So you have to give that details to your observation so that it will be included as part of your findings, no, nga siguro that would be relatable to your findings as well. Now, maybe this is not just about the human intervention, but also kanang, let's say, factors would also affect the gas, the oil and grease coming from the yacht, no? So, inana. Another is consider possible sources of variation by looking at the different land use practices and activities. So, example in rivers, if the river itself does not clearly show that it's definitely river alone, but many factors such as let's say kanama people residing nearby the rivers may also affect the quality of water. So you also need to do or consider possible sources of variation. Maybe in the upstream there is an agricultural area that would definitely carried off by runoffs to the nearby bodies of water, which is the river itself. Then another is siguro sa midstream, for example, of the river, the area has a nearby poultry, no? Kanang mga anak poultry farm. So definitely, this would also give maybe give another factor or source of variation to your study. So in anak is how you identify sampling sites. So another is collecting samples and pre-treating them. So before we go over to the biological species that we are trying to deal with in terms of coastal and even freshwater ecosystem, now let's review or recall plankton. So we had this, no, in your bio, zoo. Now, I think we discussed this for those who are or who were under my class during zoology. Zoology, we don't have. Yes, zoology. Okay, so plankton again is a word that comes from the Greek, no, as defined as drifter or wanderer. So meaning, bisan asa ni sila, naa sila, no? Depending on the quality of water. So an organism is considered plankton as if it is carried by tides and currents and cannot swim well enough to move against these forces. So these are measured in micro, no? Yes, in micro or microns, because they are too small to see with your naked eye. So these plankton can only be seen in or under the microscope. So this plankton, or some of the plankton drift their way, for their entire life cycle. So others are only classified as plankton when they are young, but they eventually grow large enough to swim against the currents. So plankton are usually microscopic. That's what I'm telling you. And they are often less than 1 inch in length. but they also include larger species like some crustaceans and jellyfish. So what I'm trying to say is that during the life cycle of the crustaceans, no, and jellyfish, some crustaceans and jellyfish, no, I mean, their eggs, or let's say larva form, is as small as the plankton. But when they grow big, no, they're no longer called as plankton when they grow big. So, muna sila, tinatawag na to na sila, part pa na sila as plankton when they are still juvenile. So, let us also recall benthos. Benthos or benthic organisms live on the ocean floor, either on the substrate or inside it, buried or burrowing in the sediment. And of course, kani sila na natay ginatawag na two types of benthos or benthic organisms. These are what we call the epifauna and the epiflora. So for epifauna and epiflora are living on the ocean floor, either on the substrate, no? Sa mga bato-bato dayon kanabitang nagpilit-pilit na sa mga bato. So these are what we call, or nagnabuhi na sa mga bato-bato. These are what we call the epifauna and epiflora. Another kind we have is the infauna, which are inside this kuan kaning let's say kaning mga epifauna and epiflora. Maybe they are buried or maybe they are burrowing. So example na ng mga infauna is kanang mga hermit crabs, no? Na supposedly hermit crabs don't have their own shell, so they live by other shell in which they will be using it as their source of shelter and protection. So, kani sila maginatawag na to ni sila ng mga infauna, which can be found inside the epifauna and the epiflora of a benthic organism or benthos. So, kana sila, no? Benthic organisms also may be sessile or kuan, kanabitang dili sila gamove. Yes, and attached to a firm surface such as the rocks and the man-made structures or mobile. Pwede po sila, nagalihok po sila, no? Moving freely on or in the bottom sediment. So basically, benthos are present in all habitats from the intertidal to the abyssal plain. So, gikan sa kung asa ang balod mo igo, mahuman dayon dito dayon sa abyssal plain sa lalum nga part, they are all present. So the abundance and diversity decrease with increased water depth, so which means that the distance from the shoreline, no, has a declining species, no, or decreased diversity and abundance, no, from the shoreline. So meaning mas gamay ang population sa benthos or benthic organisms kung magkalayo na or magkataas na or magkalalom na ang tubig. So as a result, magana siya because of mga dwindling of food availability. So which means that if there is a lot of food nearby shoreline, then definitely they are existing or they are increasing in population. But away from the shoreline, gamay raman ang ilang makaon. So definitely some may survive, some may not. No? And aside from that, due also to the factors such as increased pressure. So the water depth, meaning kung taas na kayo ang tubig, it will increase also the pressure and of course decrease water temperature. So increasing pressure, decreasing water temperature, so which makes them uneasy to live within those kind of habitat, no? Habitat. So familiar examples of benthic organisms include macroalgae, sea grasses, no? Corals, barnacles, mussels, sea urchins, and sea stars. And benthos may be autotrophic, such as the sea grasses and algae, and can also be heterotrophic, which preys on other organisms. So this is one of the background of the benthos. So now let's focus first on the plankton community. So these are, now as you can see, these are the size category or the classification of plankton based on size. So we have what we call the ultraplankton, the nanoplankton, the microplankton, the macroplankton, and the megaplankton. So these are their size ranges. Now, so as you can see, the megaplankton is greater than 2,000 microns. So as you can see, we have this in our EST laboratory. So this is a plankton net in which that this would also what do you call this? There are different types of plankton net. No, so ang iha ani, it depends on the mesh size of this type of plankton net. No, whether what specific genus are you focusing? So if you're more on microplankton, so definitely your mesh size would also be 200 and below microns, no, ang iyang size sa iyang net dari. So now, what is the procedure of getting this plankton community samples? No? So first is what we call the bottle sampling. You need to prepare enough 1-liter bottle with a narrow mouth. Of course, when you use your nets, dako manggod kay to siya. So you need mouth wide mouth nga mga polyethylene nga mga gamit. And then you collect 1-liter sample from a specific depth. So basically, plankton manggod community is very crucial, no? So when you decide to deal with plankton community, you have to consider sampling time. Kaya kaning mga plankton community, they can only be present, no, during sunrise and sunset. So not really sunrise nga, medyo late na kaayo, like 8 o'clock maybe, that time, 8 to 9 maybe. Mo lang na sila nga magpakita, mo to sila sa shallow depths of the water or sa surface water. But tungod kay mag-init naman, no, taas naman ang temperature about 10 a.m., 11 a.m. So definitely they go, no, that's what they call vertical nga kuan, to call this vertical migration, I mean. So when this plankton community, no, reaches surface, so magstay sila diha for short period of time. So when they sense that the temperature is increasing, then definitely they would reside to the lower depths of the water. So kaning mga for example, zooplankton, no? I myself has been dealing with zooplankton community since college. So my college thesis is all about zooplankton, and even my master's thesis is all about zooplankton. So I can say that I'm maybe a little bit knowledgeable about plankton communities. So specifically zooplankton, because we have two different types of plankton communities. So we have the phytoplankton and the zooplankton. So from the word phyto, it deals with the plants. We're more on plant lang ang ilang based. So this another kind, no, zooplankton, which eats phytoplankton. So these are much bigger than the phytoplankton. So ginakaon ni sila sa zooplankton ang phytoplankton. So another is that yes, another movement of this zooplankton community, no, is during the night. So mas daghan pud sila, medyo dili kay sila daghan during gabi igit kaayo, but pag kuan na siya sunset, sunset. The beginning of sunset, that's the time that they vertically migrate to the surface or to the narrower depths of the water. So mao na siya nga kung daghan pud zooplankton in that particular area, then definitely there will be more population of fish, because it's the only yes, it's one of the sources of their diet, ang kanang zooplankton and even phytoplankton. Yes. So another, no, yes, that's why we emphasize, no, specific depth. It's because it depends on your objective, because there are zooplankton that can only be seen in the surface area, but other species can only be seen in the larger depths, like 100 meters. But if your capacity, no, or your net's capacity is only in the surface area, because when you do depth or strata or by stratum, then definitely you'll have a bigger vessel with you. So kung magsampling mo na mo'y baruto or na mo'y dako kaayong barko, tas bugat pa din na kayo nga plankton net, dako kayo nga plankton net yung gamiton. So inana siya ka crucial. So another procedure we have is preserving of samples by adding borax buffered formalin. So this borax buffered formalin, no, gives the concentration nga dili siya acidic in terms of its pH. So this borax buffered formalin, yes, this borax makes the formalin not to be acidic, because if it's too acidic, it will cause makuan siya, no, ma-distort ang sample or mabungkag ang sample ng mga zooplankton kaya nasubraan siya ka-acidic. So the water and the formalin, or the borax buffered formalin to the water sample, is within a ratio of 1:9 ratio. So kana, no? And then of course you have to label the bottles. So in labeling the bottles, you need to be more specific. So for example, you need to do what particular sampling site, what time, kano saan mo siya gi-sample, gikuha ang sample, and then simply you need to also note kay dili man kayo ka makanote ano ka mabasa man imuhang bottle, you need to buy parchment paper so that imuhang gipang sulat dito nga information is dili siya ma-erase. Kaya usually in ana, no, basta water, it deals with water, your label will get wet. So one thing that my professor told me about collecting samples on marine, yes, marine coastal. And if you're dealing with zooplankton, then you have to make use of the parchment paper with a Mongol 2 pencil. Kaya para dili siya ma-erase yun, bahala pag acidic or dili ang kanang water na gibutangan sa imong sample. So another procedure, no? Continue, let's continue. Let the sample stand for at least 5 days. So that no nga katong mga samples nimo nga buhi pa ana sa kuan kay usually when you sample them, you can really see that they are kanagitang swimming sa imuhang bottle nga medyo duulon lang nimo no, medyo makita kay nimo nga naay mga puti-puti nga ga-swim-swim, kay buhi pa sila. But since you've already treated them in borax buffered formalin, then definitely you still have to to leave them for 5 days so that this species will also reside at the bottom of your narrowed narrowed mouth nga kanang mutanganan kay para moadto sila sa ubos, no. And then pwede nimo siya kuhaan ng tubig every now and then so that it will be easy for you to identify the samples or transfer these samples to another bottle. So inana. Next is transfer to a bottle of smaller diameter and let stand for another 5 days. Decant again until there is only 50 ml. So unang gingon, no? So pag mastanan mo siya 5 days, so mo di man mulutaw ang mga zooplankton nga patay na naman sila sa ubos. So nasa mo settle sila sa ubos. So kailangan mo sa i-transfer in a smaller diameter and stand na po nimo sila for another 5 days para madto na po sila sa bottom. And then decant, when you say decant, you reduce the amount of water in your bottle, which has a smaller diameter, then hantod 50 ml na lang din ang mabilin sa water. And then after that, with the use of microscope, and then you'll finally or you can identify it by yourself by dissecting the zooplankton, then you identify the sample and name it. Kaya para next nimo nga pag-sampling, diretso na dayon ka mag-count. So you don't have to do another identification na po. So less hassle. So there are different kinds or types of plankton net. So we have the vertical tow, in which it is used usually in fresh water, which varies from 50 to 70 microns. So as you can see, when you do vertical tow, ang iyang method is, of course, vertical ang imong pagkuha. So for example, you have the plankton net. Let me show you. For example, you have the plankton net. So ang imong buhaton is, imo lang siyang ihulong. So for example, this is the plankton net. Ihulong lang mo siya sa ilalong vertically. And pwede dahil mo siya ipaangat dayon. So that's what we call the vertical tow. So sulod lang siya because the thing, if you can see here, the plankton net, no? Kasi sa plankton net na siya yung murag funnel dara. So ang funnel nga ibutang dara, no, receives the amount of water you collected during your sampling. So tanan nimo ang nadakop dara, moadto din sa ilalom dari, then dana nimo makuha ang mga samples nga imuhang na imong nakuha, no? Or imong nakuha sa sampling. So the total volume filtered is equal to area of plankton net opening times the depth. So for example, if you measure it by vertical tow for 50 meters, then definitely you'll multiply it with 50 meters. So vertical tow, vertical. Diretso lang siya pababa. But in case-to-case basis, no, this is not actually useful to some of you who don't have any vessel. Kay we will be needing vessel magod kay dapat bugat ang nasa ilalom para mo kuan siya mo ato siya sa ilalom kay usually kining mga plankton net dili kaayo siya mulutaw siya, so it needs a lot of force underwater for it to sink, and that's how vertical tow is being applied sa inyong sampling. So muna siyang ato ang i-consider. Another is, of course, the horizontal tow. So for horizontal tow, no, it's all about dragging a plankton net at a given or desired depth. So you will tow for about 2 minutes. So here, all you have, this is very convenient for those who can't really afford renting a vessel because it's very, very costly, manggod. So usually in horizontal tow, for example, this is your plankton net, imuha ra siyang i-ana sa water for 2 minutes. So, bale, dili ninyo siya makita, continue towing for 2 minutes, and makita na nimo siya visibly if naa na siya sa imuhang container, nga katong 1 liter container. So, ano mo siya at certain depth, maybe if makaabot ka imuhang kamot or ang imuhang string dari for about let's say 1 meter, then it's good. Okay na po ito sa surface, so it doesn't matter, at least naa siya at the shallow depths of the water. This is applicable for shallow depths of water, horizontal tow. So imo siyang i-tow, i-ana, or i-drag lang nimo siya, no? So that's how you capture or do sampling in terms of horizontal towing. So let's now move on to the benthic community. Benthic community, no? These are organisms inhabiting the sediments of freshwater bodies. So as you can see, we have the microbenthos, meiobenthos, macrobenthos, and megabenthos. So, yes. Okay, going back, no? So these are the classification. Now we have the microbenthos, which are smaller than or less than 0.1 mm or micrometers, which include the bacteria, diatoms, ciliates, amoeba, and even flagellates. The myobenthos is also called as myofauna, which are small benthic invertebrates that live in both marine and freshwater environments. So we have the microbenthos, which are, aside from that, no, kaning myobenthos, those animals that are retained by 0.1 to 1 mm mesh sieve. No, these are myobenthos. So they are commonly found in sand or mud. Then for macrobenthos, you can see that this consists of organisms that live in the bottom of the water column and are visible to the naked eye. So it's all about, it forms larger than 1 millimeter or let's say 0.04 inch, which are dominated by polycate worms, pelecypods, anthozoans, no, kana sila. And of course, the megabenthos, which are greater than 1 cm. So examples of megabenthos assemblages, no, are the oyster reefs. We have the mussel beds, which also create biogeochemical hotspots for the organic matter. Then we also have the mussels, the cockles, the oysters, and even sea cucumbers, which are part of the human diet. So these are what we call the megabentos, and these are the classification of the benthic community. So there are different types of how this benthic community are being sampled. So we have the core sampling. So this is about collecting sediment samples using a core. So I think we don't have this type, but we have a core sampling in our core I think apparatus for the soil, but I'm not sure if it's okay for sediments because it's sediments are filled with or being surrounded with water. So kato siya is bigger man gud na siya part na open kayo siya. So basin pagtangtang sa sediment, basin pagtangtang niya sa tubig, mahabwa tanan ang sediment. So kani siya kay covered man gud siya from uh, all around kato kay naay part dito sa iyahang kuan nga open siya tanan. So I don't know if it's applicable as core sampling in sediment. So another is transfer the samples into labeled sterilized containers. So after sampling, after collecting sediments, you have to transfer it. And then place samples inside an ice chest within 24 hours to, of course, preserve the sediment so that you will be able to know what particular species have you captured in your core sampling. So mo na siya nga ginabutang mo siya sa ice para ma-preserve pa tong mga species, benthic communities that can be seen in this core sampling. Another is the graph sampling, which is a mura siya clip, so it's a box-like contraption with lids that open like jaws. So can collect samples from a lake or a large river, and after collection, sediment samples should be transferred inside a plastic bag. And then preservation of samples involves adding enough borax buffered formalin with rose bengal. So definitely here, the graph sampling, if you know how to use bigger scissors, it's like in ani siya, mura bitaw nimo siya kana bitang murag scissors nga dagko na you have to use both hands for it to kana magtagbo siya. So kana dayon you'll face this in the lake or large river, and then you can grasp sanodine or sediment samples. So tabunan na niyo siya, ibalin mo siya sa plastic bag, and then you have to preserve it using the formalin, borax formalin, to preserve the species that you have captured. So another is the graph sampling. So in the laboratory, remove the fine sediment through the filtration using the 0.5 mm mesh size metal sieve and preparation of the myobenthos, macrobenthos, and megabenthos. So of course, in grab sampling, all you have to do is to grab. Ana lang. So when you talk about grab sampling, you have to do grabbing. So mostly, ikaw kung makita nimo siya, then definitely pwede nimo siya i-grab. Or maybe if you have a kanang sieve, then you found this particular species in the sieve, then you have to separate it whether asa siyang classification na belong. It's either if it's myobenthos, macrobenthos, and even megabenthos. So that's it. Another type we have here on how to get samples, in terms of Bentic community, we have dredging. No, it is a bag-like sampler that with a metal rim, and then in collecting samples, attach a dredge using a strong ropes at the posterior end of the boat, and then samples collected are processed in the same manner as in graph sampling. So, in iyang buhatan ani is, ang iyang kuan lang is, this is very simple, no, dredging. But I don't know if it's still applicable nowadays and even safe. Okay, what if your sampling area has a lot of coral reefs, no corals or coral communities? So definitely it would devastate the area. So inanagya siya also kani ang kuan barko, then imo lang siyang dagan. So definitely mabangga to niya itong mga na coral if ever magani na coral dira na area. So not really advisable, no? Though it is very convenient if you want to grab, I mean, yes, to do sampling, kay diretsura siya nga mosulod dito, ikaw dayon as researcher, igo na lang ka mag-separate dayon once you already collect yes, samples coming from dredging. Another is the artificial substrates. So usually, this is used to determine the benthic community by choosing the appropriate substrate for the organisms you would like to study. So definitely not all artificial substrate is made by this one, is made with this kind of material. So definitely, if you have plastics, you can still use that. Dayon, depending on the mesh size, because this is too large, so definitely it will capture bigger fauna, no? So dapat depende kung sa inyong scope sa inyong study or unsa ang inyong objective sa inyong study nga makuha nga you are focusing on what kind of benthic community with each. Okay, so another is the aquatic macrophytes. So these are plants found in littoral zone of the lake, in riverbeds or in riverbanks. So usually no macrophytes, I haven't deal with them ever since, but these are usually found in lakes and or riverbeds or riverbanks na medyo magian lang siya og water, so usually maybe found in on the sides of stones maybe. So in lake manggod kay it's not moving, so kuan lang siya, wala kayo siya oxygen, so mainly ang magdepende to nga mga species is of course in terms of temperature, ganahan sila adto sa river. So usually kani mga macrophytes can only be seen in the lake riverbeds or riverbanks, wherein dili kaayo sila exposed to intense I'm not really kuan kanabitang si tawag ani dili kaayo daghan ang population within anang area. So kung lake for example, if you can see lakes, no, if you've noticed them, clear kaayo ang water, but it doesn't mean the quality is very kuan good. So kung lake gani siya and then it has clear water, then definitely it's a dead water. Itungod the quality of water there, no, na siguro mabuhi dara pero gamay ra kaayo. So wala pa siya kuan, dili pa siya moving, kay the more the water moves, no, the more oxygen it has. So kung bulaon siya, kung naay bula present, then definitely naay isda. Like in the river, no, mas makita ninyo dito nga flowing ang water, even dagat, nagin na siya presence or nagin siya life forms. While in the lake, which is still water, we call that still water, dili siya ga-move, dili siya flowing. So gamay ra kayo ang species nga makita dara. Okay. Next is the periphytic algae. So if we can still, I don't know if we've took this up. No, we took this up in our zoology, but I think I haven't introduced this. But I also have a study which was conducted by my advisee last 2017. They were batch 2017, I guess. 18, I guess. So periphytic diatoms are used as indicators of water quality because of their ecological tolerances or preferences to environmental variables and are thought to be predictable. So kaning mga periphytic algae, mura pa din sila mga algae bitaw which causes eutrophication. But they are different in terms of their species. They are also called periphytic algae. There are different families that belongs to the periphytic community. But ang focus niya ani is that when you deal about periphytic algae, no, the water quality involves more of the nutrients because without the presence of nutrients in the water like nitrates and phosphates, then definitely they will not be present in that particular area. So, yes. So the procedure of doing sampling on periphytic community is first on-site collection. So for quantitative study, it measure an area on submerged object that you want to study. So pwede nyo siya nga magkuan ka dito, mura o mag quadrat or transect maybe. So in that particular transect line, magbuhat ka dito og mga plotting. Like for example, for a while. So for example, this is your transect line. You will be doing sampling plots within the transect line. So this is, for example, in your 50 meter or let's say 60 meters, you have the distance you have a 10 by 10, ana, dayon ka na. Ang iyang distance, you measure the distance between this sampling plots, kana siya. And then another. So, and kung mautana mo, no, basically, when you do sampling, no, you have to do 3 lots or 3 trials because it's how you get the average of kanabitang reliable gid siya na result. So that's why you do 3 sampling sites or subplots and 3 trials. So that's it, no? For ano, no? Use bottomless polyethylene bottle. So kani siya still for quantitative study, no, you'll do bottomless, so meaning ipastay lang mo na siya within a week siguro or maybe in a month. Then usually kung river manggod, di ba, for example, this is the bottle. Ana siya. Tas ang water current kay di ko kabalo mabuhat og bottle. So dapat dari nga area is empty siya. I mean bottomless. It's because you let the water current no enter here. And then pagkadugayan ana mura man na siya og muligyas, di ba? Ang kaning sitawag ani ang bottle. So definitely mag-build up na dayon siya og mga periphytic algae inside, which will which makes this bottle their new habitat. So ang water current mosulod dara nga magligyas na siya, and then na din sila magtapok sa ilang kuan ng population. Yes, can be. Dayon, we also have for qualitative study, brush the area first and then scrape the object of interest. So, for example, if the area in the river is basically stony area or rocky area. So this is the stony area here. Tapos usually makita mo niyo na naiuban dira nga green ana. Naiuban ana ga green na siya diha. So all you have to do is to brush the area and then scrape din ninyo siya. You brush it first because to smoothen the surface and then you scrape it. Kaya para dali ra siya tangtangon ang katong mga species that live within this kind of habitat. Because these periphytic diatoms na can live in different microhabitats. So it's either on the surfaces like kuan kanang bato, the rocky kanang perminti siya magian og water dayon maglumot na siya, so they are actually present on that part. Tapos kato da yung mga walls, sidings no sa example sa river banks no nga nay mga sidings ana na green na, you can also use that because they are also present in that habitat. So another is procedure of the artificial substrates. So as you can see here, this is a plastic made na gikuan siya, hikot siya sa mga batuan. Kaya of course, when you talk about river manggod, definitely anon yun kaayo siya. Kaya wata ka ba umuulan, kusog. So maanod ang imuhang artificial substrate. So as you can see, with this artificial substrate, all you have to do is to make a substrate wherein pwede siya maagian sa tubig, then definitely pwede na siya mapuy-an o habitat nila. So kaning mga green dari nga murag lumot, this is definitely their growing population, the periphytic algae in this kind of substrate. So slimy na kay na sila pagawidan nimo. Then all you have to do is use microscope slide to scrape on metal sheets para makuha din ninyo. Diretso na dayon siya identify under the microscope. So that's it. Fish community. So let's now move on to fish community. So first is when you deal with fish community, inyo sang unahon is you interview, especially the fisher folks, the fishermen in that particular area. Kay para makabalo mo kung unsay nga mga available nga mga isda ang naadiha, unsay naa ilahang mga ilang usual nga gakadakop. By that, before identifying those, you'll have also to study the names of that particular fish para you'll be able to know the different physiological structure, physiological system of its body, then its capacity, maybe edible or non-edible, bani siyang kind of fish. So, if you want to deal with fish community manggod, what is the purpose of studying fish community that is not edible? Kaya kung for example, mag-research ka isda, anong gina-research mo na nimo? Wala lang. Diba? Dapat ang imuhang main yun nga idea is pangitaon to nimong isda nga edible because it can actually be eaten by the locals, and then you'll be able to know whether this kind of fish community can be a source of poison, or maybe it can absorb a lot of heavy metals, no? So wala ta kabalo kung kani siyang isda ginakaon ba ni siya or dili ginakaon, no? So you have to do interviews between fishermen, vendors, and even fish traders, no, to assess the different kinds of fish present in surface waters. And then um, names of fish vary depending on the locality. So if natay ginatawag na common names and local names. So when we say common names, it is widely used as that kind of name, no, sa iyahang kuan, that kind of species, for that kind of species. But when you say local name, then definitely lahi siyang pangalan. Okay, for example, uh, common name for yes, kani bitang bangus is mudfish. Pero here, it's a local name, bangus. Pero sa uban, we don't know what's their local name. So depending na sila sa locality kung si term nila. And especially the dialect would also affect to the local names of this particular species. So aside from that, no, identify relative abundance and seasonality of fish. So maybe they are abundant in that particular season, like for example, the wet season, like from October to uh, March, but when it reaches April to let's say July or June, yes, April to July, then definitely, nana, no, seasonality. So it doesn't mean that if you found a particular fish, it will be present, no, throughout the year. So definitely magdwindle ang ilang population because they still have to do spawning. When you say spawning, no, it's their mating season. So definitely, if it's their mating season, they are not active. So kailangan nila magpadagan silang population. So that's how they replenish their population. So another procedure we include, no, is the market surveys. So if fishermen, no, or the locals are not or has kanang low data about the fish population in that particular area, then definitely go to the nearby market, no, in that area so that you'll know what different types of fish species are available. So that's what we call for rapid appraisal. No, you can visit makeshift markets or talipapa so that you can be able to know those fish species that are available in that area. And then fish caught from open waters are seldom brought to the main market nor to the major landing sites since only small quantities are caught. So this is also one of the considerations you might be you might want to deal with. Kay usually kung makakita ka nga, if you want to deal with this specific kind of species, you also need to know whether this population, no, the population of this species is vast. Daghan sila. So, usually kung makadakop man ang mga fisher folks ani, tapos gamay ra kay nga population or gamay ra ilang nadakpan ana nga kinds of fish, lahi na nila. So, ang mahitabo dili na ninyo makita sa market. Kay gamay raman kayo, di man sila mahalinan ana, di man na siya paliton kay mga dagko man nga quantities ang ganahan nga ibaligya sa market, no? Okay. So syempre for profit purposes. So another is try to take photographs if you are not familiar. So this is the way of kanang giving or identifying easy way of identifying this fish community. So aside from taking photographs, you don't only take photographs na murag selfie lang nimo ang isda, but you also need to do measurements. Like when you take photographs of fish community, then definitely you'll have to bring with you a caliber or um, ruler so that you'll be able to know the length and the width, no? Dili lang na siya length and width, but also the fins, the color. No, you have to do that on your observation. So that is for the procedure of fish community, no? So that's for basically abundance and distribution. But when you deal with other studies such as heavy metal in fish, no, we had that during 2018, I guess 2017. So I had my students with me who worked on heavy metals in fish. So, na-present sa andahaw, sa lapu-lapu. So, definitely, this fish, no, population, these fishes are very, kanang dangerous nowadays. So, dapat lesson na ang pagkaun-ana nila. No? So, we also want to know whether this fish may be affected to the nearby, um, nearby communities. So whether it may have been affected due to the establishment of industries or maybe the ongoing construction of a coastal area, baka, or maybe from residential areas or gasoline stations, which contributes to the increase of heavy metal contamination in fish. So, kailangan po na ninyo siya i-take note. So, I guess that would be all for our part 3 about biological sampling of the environment and its sampling method. So I guess that's all for midterm, and hope you're prepared for your midterm since this will be an output based. So thank you very much for listening, and we'll see you after midterms for our next video lecture for the finals. So thank you and God bless you all.